Hi, my name is David, and I am presenting on the paper Genome-wide Association Study of a Varroa-specific Defense Behavior in Honeybees by Andreas Sputter, Pooja Gupta, and Caspar Beinfield of the Institute for Bee Research and Manfred Mayer and Norbert Reins of the Leibniz Institute for Farm Animal Biology. In order to understand the topic and question being addressed in this paper, you will need to have some background information. First of all, Varroa destructor is one of the most damaging parasites to honeybees. It is a parasitic mite that attaches to the body of a bee and sucks its fat bodies for energy. This severely weakens the bee. The female mite enters a honeybee brood cell, a cell housing a larva, and lays its eggs as the larva enters a pupa stage and the cell is capped by worker bees. The young Varroa mites will then emerge from the cell with a young bee and spread to other bees and larvae. Varroa mites are also potential vectors for devastating viruses in bees. You can see a Varroa mite here, shown on the slide. In this paper, a genome-wide association study was done to identify potential genes responsible for hygienic behavior, a behavioral response in honeybee workers to uncap and remove Varroa parasitized brood. This behavior is a normal response to many brood diseases. However, most bees are unable to detect Varroa-infected brood. Less than 1% of worker bees are able to detect Varroa in brood cells. Understanding which genes are responsible for this behavior can help future attempts to breed Varroa-resistant honeybees. The image on the slide shows capped brood cells on the right and uncapped brood cells on the left. Genome-wide association studies are a method of associating genetic variants with certain traits. The genomes of many individuals are scanned in order to look for genetic markers which can predict the presence of a trait. In this case, the trait is a hygienic behavior in honeybees in response to Varroa-infected brood cells. This is a simplified representation of the methods of the study. First, in order to have a diverse and large sample size, the researchers took 10 queens that have been bred for Varroa-resistant behavior and inseminated them with pooled sperm from 250 unrelated drones. The offspring were phenotyped for hygienic behavior in response to Varroa destructor by introducing 2,000 workers into a colony with 170 brood cells. 45 of which were artificially infested with a single Varroa mite by cutting a slot and inserting the mite for resealing, 75 of which were untreated, and 45 opened and sealed as a control group. The bees had numbered tags on their thoraces, and their behavior was monitored and recorded by infrared sensitive tags camera under infrared light to not disturb the bees. After 8 days, the bees were killed and stored in ethanol. This was repeated 11 times for a total of 22,000 offspring phenotype. The recordings were scanned for bees demonstrating detection and uncapping of Varroa parasitized brood cells, and the top 122 performing bees were selected for genotyping, and an equal number of bees which did not display any hygienic behavior were selected for genotyping as a control. DNA was then extracted and SNP genotyping was performed using an Affymetrix 44K SNP array containing 32,632 SNPs, and the data was analyzed. The authors tested each SNP for association versus no association and created this Manhattan plot showing minus log p values for each SNP. The SNPs with a minus log p value above the dotted line, representing the significance threshold, show significant genome-wide associations with detecting and uncapping Varroa-infected brood. These SNPs appear at chromosomes 2, 3, 5, and 6. And here is a chart showing the individual SNPs that were identified. Blast searches were carried out to identify candidate genes. Genes that were nearby the SNPs were considered, and from the search and functional evidence, 4 candidate genes were selected. The candidate genes were adenosine receptor, cyclin-dependent kinase 5 activator, octopamine receptor beta 2R, and odorant binding protein 1. So, the authors of this paper used a genome-wide association study to find SNPs that were associated with detection and uncapping of Varroa-infected brood cells. They were able to identify 6 SNPs, and from that they found 4 candidate genes. The 4 candidate genes listed here may be related to hygienic behavior in bees. The adenosine receptor is responsible for normal cellular function, but the authors hypothesized that the receptor may affect learning and synaptic function in bees. Cyclin-dependent kinase 5 activator is an activator of kinase activity in neurons and is essential for neuroid growth. This gene may affect the sensitivity of the bees to the external stimuli produced by a Varroa infestation. Octopamine receptors carry out functional roles including the modulation of sensory inputs and memory and learning, all which may be important in the Varroa resistance of honeybees. Finally, the odorant binding protein 1 possibly affects the olfactory power of honeybees and may play a role in identification of Varroa-infested cells. These findings are important because it helps researchers gain a better understanding of this hygienic behavior and how it functions to resist Varroa infestation. From this study, potential genes that influence this behavior have been identified, and hypotheses about their potential roles in this behavior were made. However, further research into genes involved in detection of abnormal brood is needed, because there may be other genes that affect this trait. There still is not enough evidence about which genes are responsible for this data to be used in breeding Varroa resistance in bees. Until more research is done, monitoring bee behavior will be the most important measurement of Varroa resistance in bees.